Thank you. Yeah, so I'm going to be talking about inflammation, which is one of the hallmarks. It's the second generation hallmark of cancer, not an emerging hallmark. It was there in the middle. So a lot of my research is focused on sort of dividing the immune response into pro-tumor and anti-tumor effects. And then sort of focused a little bit in this work on just the tumor promoting side and what is the consequence of inflammation as it has to do with actually concomitant resistance and metastases. So this is the general slide I like to put up where I say, I'm considering the dual actions of the immune response. These are the anti-tumor effects, from macrophages, cytotoxic T cells, etc. We're not going to consider that in this talk. We're going to focus more on the pro-tumor actions from the, the M2 macrophages, the Tregs, etc. And I understand in the literature it can be confusing sometimes when biologists say inflammation, they are referring to sort of the initial influx of these types of cells. And this is what happens in acute inflammation, but in chronic inflammation, which is what is in cancer, then we're sort of focusing on this side, which is the sort of slow burn, never resolving type of inflammation. So again, I'm considering in this talk the pro-tumor actions of immune cells that immune cells initiate wound healing process. They promote angiogenesis, invasion, lymphangiogenesis, matrix remodeling, and it's generally associated with a lot of bad things in cancer. This is a nice slide I like to talk about where there's a bunch of different sources of information. So where's the inflammation coming from? It can be, environmental or dietary induced inflammation, therapy induced inflammation, chronic inflammation or autoimmune symptoms, and tumor associated inflammation. So the tumor itself can be driving this chronic inflammation that all sort of leads to the evolution of the tumor into a more aggressive state. And so, yeah, so there's the difference between acute inflammation, which if you go into the literature, you might get confused because the inflammation can mean two different things, but we're going to be talking about chronic inflammation, which is this sort of ongoing, sort of never resolving immune response that is generally doing pro-tumor actions. And when tumor cells die, or when any cell dies, if it's apoptotic, it's generally a clean death, right? And so this does not result in inflammation. But if you have necrosis and necrotic cells, as we have heard this week exists in tumors, then you get the release of these damage and danger signals that promote the pro-inflammatory, chronic inflammatory state. There is some hope. Inflammation does all this bad stuff, but we do actually have already pre-existing drugs that target inflammation. Of course, non-steroidal NSAIDs, non-steroidal anti-inflammatories, and more specific targeted drugs to target inflammation. One of my research areas is on cancer cachexia, which is linked with inflammation and driven by cancer. And so that's why I put it on the slide, because I like to always talk about it. But it is another consequence of having this chronic inflammatory state is that, it leads to decrease in quality of life. The patients start losing muscle mass and fat deposits, increase in pain, decrease in the ability to sustain treatments, and ultimately mortality. Right, but if we use anti-inflammatories, then we have the potential that we can slow cancer progression, we can reduce cancer risk in general. Some older folks you might know take a daily aspirin. That's sort of like a preventative thing that's trying to slow the ultimate cause of inflammation. So in this talk, I'll be focusing on a COX-2 inhibitor, which I was actually kind of excited because I recently had a surgery and then they gave me celecoxib, which is a COX-2 inhibitor, and I actually knew what it was, and it was like two worlds colliding for a little bit. But yeah, COX-2, celecoxib is a drug that targets COX-2. And so basically, what happens is if you have COX-2, then through all these physiological effects, you get the promotion of angiogenesis, the promotion of proliferation. You shut down anti-tumor immunity like CD8 T cells, and you shut down apoptosis. But by blocking COX-2 with anti-inflammatories or celecoxib, which is a target specific to COX-2, then we hope to break the pathway and allow anti-tumor immunity and stop proliferation, basically. So here's an example that sort of wraps the modeling work up, and that is breast cancer. So there's the idea of concomitant resistance in cancer biology, where a primary tumor might be imposing some kind of inhibitory signals on the metastases. And this is an example from breast cancer. So if you do surgery on the primary tumor, then the relapse risk generally has two bumps. And the first bump is thought to be sort of the awakening of metastases due to removal of the primary and the cause of the surgery, like the initial the trauma of having the surgery itself, but also the removal of the primary tumor. What was the primary tumor emitting that might have been holding the metastases small? And then the second bump is more due to, stochastic effects, accumulative mutations, et cetera. So it's less targetable for treatment specs. So getting to the idea of concomitant resistance, this is a mouse data that I found. And so they kind of broke it up into three groups based on how big the tumor is. If the tumor, the primary tumor is small, but bigger than a small metastases, then generally the fact that metastases is kept small is thought to be due to immunity. So the immune system has somehow been primed enough that it can hold the metastases small while not being able to control the primary. If it's sort of a middle-sized tumor, then it's thought to be an anti-angiogenic inhibition, where this tumor is releasing anti-angiogenic properties or factors that are limiting the growth of the metastases. And then in larger tumors, it's thripsia, which is more like nutrient availability and those sort of ecological arguments, like this is eating up all the glucose, so there's nothing left for this one to grow. Now, if you, yeah, yes, it's disruptive. No,
it is okay.
No.
So why would the primary tumor do angiogenesis inhibition? I mean, usually.
Well, so the tumor cells are producing both pro and antiogenic factors, and so the idea is that pro-angiogenic factors are short-lived, high clearance, and anti-angiogenic factors are low clearance rates. So they have the ability to, flow systemically and inhibit the metastases. Yeah. Now, if you imagine cutting out the primary, then you release all those inhibitory factors, right? So then that would lead to the growth of the metastases. And so one of my questions is sort of what role can inflammation play in this concomitant resistance idea? So here's the model that we have cancer cells with a dynamic carrying capacity and immune cells. And the immune cells are only doing pro-tumor things, so they're going to promote through inflammation the growth of the carrying capacity through, basically growth of blood vessels. But they do take up space in the environment. So we have, a logistic growth for the tumor and basically logistic growth here for the immune response with recruitment. And then this is the Honfeld model for the dynamic carrying capacity, but we've modified it to have the immune system being able to play a role in growing the carrying capacity of the tumor. And it has a pro and anti carrying capacity growth role, this is generally thought to be, the stimulatory effect of promoting blood vessel growth, etc. And this is just the fact that those immune cells are also there in the environment and they are taking up space, and they were, ultimately there is some maximum size that it can occupy. Okay, so the parameters here, A and B, are a little bit important. They're controlling the weight of the immune response in doing those pro-tumor actions. And I just imposed these limits to keep everything sort of well behaved in the model. And then we're going to insist that if the weight on the stimulation should be bigger than the weight of the inhibitory part, so A should be greater than B when we're talking about a stimulatory, an immune, an inflamed environment. So using some data, which I'll show in a minute, we do simulated annealing to fit some model parameters. We fixed these ones as best that we could, just fixing the maximum size of the immune response, the initial tumor volume due to the injections, from the experimental setup, assuming initially there was no immune system present, and fixing these ratios of the inflammatory response. And this is the fit to the data we get. So this is the average of the data points. We fit the model, and the green is the immune response. So the immune response is sort of there. It's growing alongside the growing tumor. It's growing the carrying capacity. Okay, so that is sort of the base model. Now we can explore both the model and how we can relate it back to this idea of inflammation driving the tumor growth. So this is just looking at, where in this A-B space should we live? So I said A is sort of the weight that the inflammation is doing and driving the tumor, and B is fixed on the different curves. This line is when A is equal to B, so technically then the weight would be the same in the actions. And if we're working in a pro-inflammatory, pro-tumor environment, we're generally up here. And the effect on the maximum size of the tumor is that, stronger weight of the inflammatory actions is going to result in a larger grown tumor.
And where was your equilibrium on the previous? Can you go back up?
Oh,
Okay.
just this. It's not really that, but just for the modeling sake, we're saying it's there. And then we just looked at sensitivity of our model to the parameters, of course, the ones that we fitted and the ones that we fixed. And obviously the growth rate, the stimulation rates, those are the ones that popped out biggest. And the, what do you call it? The initial condition for the tumor size was also very sensitive. So I found a second data set. This is all within the same mouse tumor model system. But this paper specifically looked at blocking the COX-2. They either had a COX-2 knockout mouse or they used an NSAID celecoxib drug that I mentioned. And so we simulated that in our model by saying either if it's a knockout, there's just no immune response. If it's an NSAID, then we said, okay, well, you do like PK things and you have some injections, etc. And so we modified it by saying the immune response, the anti-inflammatory will, decay our inflammation. And so the data is the dark colors and the simulation is the light colors. And so we tried, we didn't really play with the A and the B, but just by doing that simple thing, by saying we're either knocking out the immune response or it's inhibiting it, we got a pretty good match with the data. So this is the knockout, and you can see we don't do as good at matching data, but with the NSAID, we can match the data fairly well. And so that's showing then, this is the tumor volume with inflammation, so that's dark blue, or without inflammation, so that's the red. And so this is really showing that inflammation is the cause of this much growth in the mouse model. This was exploring the role of A and B. So if we go back to playing in that parameter space, can we better fit the data? And basically, we kind of determined that the initial place that we picked was good enough for our purposes, but we can play with A and B to try to match data better if that's something that we desire to do. So now we had inflammation in there. We thought, okay, well, what if we also play with chemo? So we threw in some pulsed chemotherapy, which is going to kill some cancer cells just by log kill. And then we can look at another data set by Meng where they did an NSAID with chemo. And this is let's see, black curve is the result of the chemotherapy. And this is, the chemotherapy drug. And this is the tumor growth with the anti-inflammatory that I believe in, yeah, this is their anti-inflammatory. That's the yellow dosing of the NSAID that was given. And so I don't have the control picture here, but it is delayed compared with the control. On the next picture, we have the control. So this is the combination that they did together, like almost to 78% of tumor reduction. So we see if you put all the results together, the initial tumor was growing this big. If you give it NSAID, you shrink it a bit. If you give it chemo, you shrink it a bit. And if you give both together, then you shrink it more. And that's just matching. Blue is experimental data, and this is our simulations that are matching. Then we also looked at anti-angiogenics. So we went back and found some data set for the same mouse model about adding an anti-angiogenic in here. I will say this is the caveat here is that anti-angiogenic therapies work great in mice, but don't really work that well in humans. So in the mouse, in our model of the mouse, we're actually able to cure this LLC tumor, but they would not translate to humans. But we modeled then the anti-angiogenic and added that in, and then you can look at the combinations as well. So what I think is interesting is, of course, all three does fantastic, but if you look at NSAID plus anti-angiogenic, that does fairly well as well. And that is in particular targeting the healthy cells, not the cancer cells, but we're targeting the environment, right? We're targeting the immune cells and the blood vessels or whatever other cells are in the environment. And that was enough in this model to clear the tumor.
Brief question.
Yes.
Because I know, was there anything about like the ordering of the dosing when you're doing that combination? Because
That's a
I
good,
did a project where we were working on like, should you give anti-angiogenics up first, then chemotherapy, or the other way around?
that's a good question. I did not play around with dose timing. I think
Right.
everything was given on the same sort of 5-day schedule. But that's a good question you could play around with. Can you improve this by changing dose
Yeah,
timing?
obviously, you need the vessels for the delivery of the chemotherapy, right?
Yeah.
Yes.
Yes, yep. And so coming back to the idea of the concomitant resistance, we have a data set where there was actually 2 injections made simultaneously, one on the left and right side of the mice. So the initial model was fit with the control data, which is red, and then they did these double injections, and they found that in all but like 2 of the mice, 1 tumor grew large and 1 tumor grew small. And that was, I think it was 8 out of the 10 mice exhibited this phenomenon. And so we said, well, can we explain this using our idea of inflammation? So we took 2 copies of our simple model and coupled them together with one, inflammatory source, and said, one is left side, one is right side, and then looked at how, they're coupled together. And this, you can't see it down here, but this is like, if the left side has all of the immune cells at the end of this, the simulation time, then that tumor will be large, and the other side would be small. So basically, that the size of the tumor is coupled very tightly to how much of the immune response is pulled to that one site. And so we did a bunch of different explorations to explain why we would have one tumor growing big and one tumor growing small. And the most, in my opinion, the most biologically feasible explanation was the simplest, in which is that we just had a pre-inflamed site. You know, the mice are in multiple mice per cage and they kind of are rough, right? So it's possible that one side maybe had a bruise. If it has a bruise, then, you know, one side has a bit more inflammation there, the other side maybe doesn't. That's going to correspond to a little bit better initial carrying capacity. And that was enough for us to explain in the model why we saw the deviation of the growth rates. Everything else in the simulation is the same. Initial tumor injection is the same, the initial growth rates, initial recruitment rates, etc. This is another data set where they also observed this sort of separation in growth rate, why you get one larger, one smaller tumor, and then they resected the larger tumor, and they actually claimed that then they saw an acceleration of growth in the smaller tumor. But I kind of argue that this is not an acceleration of growth. This is just the continuation of the exponential curve. And so we used our model to simulate that and looked at, you know, what is the growth rate for that data using our model. And you can see the best fits here if you sort of include the pre-surgery, if you're just fitting to the pre-surgery data or the post-surgery data or all the data points, the growth rates are essentially the same. The growth rate, so like looking at this number here. So there's not really an acceleration of the growth rate when you resect the larger tumor. But what is happening is that what our model suggests is happening is that you're sort of releasing that inflammatory pool that can then boost the metastasis to grow larger. So here we did the simulation where we spawned a metastasis on day 10. We did surgery of our primary on whatever, I don't know, this is day 50. And then this is how big the metastasis would have grown given our coupled model. But when you do the surgery, you boosted the metastases to be able to grow larger by sort of giving it back that inflammatory pool.
By the factor approximately 2, yes. Sorry.
Yeah, but that, I'll get to that. So, yeah, here. So this is the effect of surgery day when if we do day 50 surgery or day 20 surgery, what is the relative change to the pre-surgery metastasis volume? You know, that boost, you said it was about double, but it's not always about 50%. So depending on when you do surgery, we get this weird shaped curve that, you know, there would be an ideal time to do the surgery where we would end up with the smaller outgrowing metastases. Now, if we assume that the act of doing the trauma of the surgery actually boosts the body to produce more inflammation, so we're increasing the carrying capacity of the immune response here, then that's going to boost that effect because you're giving more fuel to drive the cancer fire. But the use of NSAIDs then can dampen down that effect, and it actually flattens out the curve and then shrinks it down. So using anti-inflammatories can sort of suppress this surgery-induced boost to the metastases. And good, I'm on time. So just to conclude, we developed this model for inflammation to look at how it can drive tumor growth and to explore the use of anti-inflammatories and other potential treatments such as antiangiogenics or chemotherapy in controlling the tumor, and explored how it could be playing a role in the process of concomitant resistance and these ideas of outgrowing metastases due to the resection of the primary tumor. And more recently, so this is work from like 2019, but more recently there has been a analysis of a study where they looked at breast cancer patients who received adjuvant chemotherapy, neoadjuvant chemotherapy, or anti-inflammatory NSAID treatment, celecoxib, for 2 years post-surgery. And they found that the chemotherapy neoadjuvant or adjuvant didn't really change the risk hazard ratio, or the risk, whatever ratio is called. But 2 years of anti-inflammatories for patients who did not receive chemotherapy at all, 10 years later decreased their risk factor by 35%. So, I mean, that's pretty good. This work was done involving my former master's student, Farjana, and was supported by Ryerson, which is now Toronto Metropolitan, and CERC. So thank you very much.